All right, welcome to The Explainer. Today, we've got a wild story for you about how an algorithm, one that was actually borrowed from the world of linguistics, is poised to completely change mRNA vaccines and medicines by solving a problem that, honestly, once seemed impossible.
So let's kick things off with a mystery that, a lot of us actually lived through during the pandemic. We had these game-changing mRNA vaccines, but they had this one huge catch. They had to be kept in an ultra-cold deep freeze, and that created massive logistical nightmares. So why? What was the deal with the deep freeze?
Okay, so the answer is pretty simple, but it's a huge deal. Messenger RNA itself is just incredibly fragile. It's like a delicate message written on tissue paper. It can fall apart really easily. And when that happens, it can't deliver its instructions to our cells, which means no protein gets made, and you get no immune response. This instability is the fundamental challenge for pretty much all mRNA medicine.
So the big goal for any vaccine designer is to figure out how to build a tougher mRNA molecule. A really stable sequence has a longer half-life, which just means it hangs around in our cells for a lot longer. And the longer it's there, the more protein it can churn out. And that means a much more powerful, more effective vaccine. The big question, though, is how do you even begin to find that perfect, ultra-stable design?
And this is where the problem goes from just being difficult to, well, almost impossible. Because designing the most stable mRNA sequence isn't just a tough nut to crack. It's a problem on a cosmic scale.
Just look at this number for a second. This is how many different mRNA sequences can all code for the exact same SARS-CoV-2 spike protein. That's a 24 with 631 zeros after it. It's a number so ridiculously huge, it's pretty much impossible for our brains to even comprehend. And to give you an idea of how mind-bogglingly huge that is, if you tried to test every single one of those designs on a computer, it would take you billions and billions of times longer than the entire age of the universe. It's, for all practical purposes, an infinite number of options to search through.
So think of this crazy web as a giant maze of possible vaccine designs, a labyrinth really. Each and every path is a different sequence, and scientists were basically lost in here, with no good way to find the single best route. Your standard methods could only ever explore a tiny fraction of these paths, leaving the best, most stable designs totally hidden and out of reach.
Okay, so you've got this impossible problem. How in the world do you solve it? Well, the answer, and this is the crazy part, came from a field that you would never expect to have anything to do with biology, the world of language. Someone had this aha moment. They realized that this massive biological design problem looked exactly like a classic problem that had already been figured out in a totally different area, computational linguistics. The science that powers things like Siri and Alexa. And this slide really gets to the heart of the whole idea. On the one hand, you have speech recognition trying to figure out if you said, "I like this meal," or something that sounds almost the same, like "I like his veal." On the other hand, you have this algorithm trying to find the most stable sequence of codons from a whole bunch of genetic synonyms. It's the same fundamental challenge, finding the one best sequence from a universe of nearly identical alternatives. So what they did was they took this really powerful algorithm from linguistics called lattice parsing. Now, you can think of it like a super efficient way to find the best path through a really complex web of choices. Linguists use it to sort through all the ways a sentence could possibly be structured to find the most likely one. And these researchers realized, hey, we can use that exact same logic to search for the most stable mRNA structure. And they called their new tool linear design. And how it works is just brilliant. First, it grabs that astronomical number of possible sequences and squishes them down into a single, manageable structure called a lattice. Then, it uses the power of lattice parsing to zip through every single possible path in that lattice at once, and it instantly finds the one with the lowest energy, which, in the world of molecules, means it's the most stable.
So the theory was amazing, but, would it actually work in the lab? Researchers put it to the test, and what they found was, wow, just extraordinary. I mean, the results were just mind-blowing. That task that would have taken, billions upon billions of years, it was done in 11 minutes. An absolutely impossible search became something you could do while you grab a coffee. And you could literally see the results in the lab right away. See, more stable mRNA folds up tighter, so it's more compact. That means it can zip through a gel faster. And that's exactly what you're seeing here. The linear design sequences on the left are just blowing past the standard, less stable design on the far right. This was real, physical proof. The algorithm was doing exactly what it was supposed to.
Then they measured how long the mRNA actually lasted. The standard stuff, it was half gone in about 4 hours. But the new design from Linear Design, it stuck around for 20 hours. That is a 5-fold increase in stability, meaning the instructions to build that spike protein were available in the cell for way, way longer.
And like we said, the longer it lasts, the more protein it can pump out. And sure enough, in the lab, the Linear Design mRNA produced almost 3 times more of that critical spike protein than the benchmark did. This showed that more stability directly leads to a whole lot more of the vaccine's key ingredient.
But okay, here's the real kicker, the thing that made everyone's jaw drop. They tested it in mice, and the antibody response, which is, the ultimate test of a vaccine, it was up to 128 times stronger. Not a little better, 128 times. That's not just an improvement, it's a complete game changer.
So, okay, this is incredible for one vaccine, but what does this mean for the future of medicine? Well, the potential here is just massive.
And the really revolutionary thing here is that this algorithm isn't just a one-trick pony. It's a whole platform. You can use the exact same design approach to create more stable and more effective mRNA therapies for all sorts of diseases, from making monoclonal antibodies to developing targeted anti-cancer drugs. It opens up a whole universe of therapeutic designs that we could never even get to before.
So by turning what was a massive, impossible problem into a design tool, we now have this incredible way to rapidly create superior vaccines for whatever comes next. This could give us medicines that are not just way more powerful, but also more stable, maybe even getting rid of that whole expensive cold chain problem and making these life-saving treatments available to everyone everywhere.
I mean, this whole story is just a perfect example of how powerful it can be to look for answers in unexpected places. The solution to one of biology's biggest puzzles was just hanging out in a totally different field, and it really makes you wonder, doesn't it? If this breakthrough came from linguistics, where's the next one going to come from?